Hi, my name is Isha Verma and I'm a researcher at the University of Michigan. Today I'm going to talk about stem cells and how these cells can be used to study brain diseases. So most of us have heard about stem cells before. We see the advertisement on the television where the doctor suggests the parents to freeze the stem cells of their newborn baby, which can be useful in case the baby has a disease later in life. Also, we see the news that stem cells have been used for the treatment of patients which have damaged corneas in their eyes. So, what are stem cells? Stem cells are like the body's raw material, which can form all the different cell types found in the human body. The most important type of the stem cells are called pluripotent stem cells, or in short, PSCs. The term pluripotent is made up of two terms, pluri, which means several, and potent, which means the ability to generate different cell types. Simply speaking, pluripotent stem cells are defined by two important properties. First of all, these cells show unlimited growth. It means they can keep on multiplying and generate more stem cells. Secondly, under suitable condition, the stem cell can form all the different cell types found in human body, such as brain cells, heart cells, and liver cells. So, where do we get these pluripotent stem cells from? One of the important sources of stem cell is the early human embryo. There are many couples who opt for in vitro fertilization for conceiving a child. There are many embryos which are generated in this process which are not suitable for implanting into the women. After the informed consent of the couple, these embryos can be donated for research purpose and they can be used to generate stem cell lines. These stem cells, as they are derived from the human embryos, are called embryonic stem cell lines. These embryonic stem cells can form all the cell types found in the human body. However, there are certain problems with the embryonic stem cells. First of all, it is very difficult to obtain human embryos and to generate embryonic stem cell line from these embryos. Also, many people consider the use of human embryos for research purpose unethical. Therefore, scientists have come up with a new method which can be used to generate stem cell line without the need of the human embryo. So, what is this new method? In order to generate the stem cell line, the scientists can collect any cell type from the adults. Most of the methods generally use the skin cell or the blood cells, mostly because the skin cells and the blood cell can be easily collected from people without any invasive procedure. To this, the scientists add what is known as reprogramming mixture. This reprogramming mixture converts the adult cells to form the stem cells which are known as induced pluripotent stem cells because they have been induced from the adult cell to behave like stem cells. These induced pluripotent stem cells or iPSCs in short can form all the cell types found in the human body. These iPSCs are very important for cell transplantation. Suppose that there's a patient who is suffering from heart disease. A doctor can collect the skin cell or blood cell from these patients, convert these cells to form induced pluripotent stem cell, and then direct these induced pluripotent stem cells to form heart cells. These heart cells can be transplanted back into the patient heart as a treatment of the heart disease. This can be useful for a variety of diseases and it is known as cell transplantation. So far, we have discussed about how the stem cell can form all the cell types in the body and in this case, particularly the brain cell. In the last few years, scientists have also come up with a method to form three-dimensional structure called organoids from the stem cell. The organoids are the miniaturized and the simplified version of the actual human organ. Here, the stem cells have been directed to form brain organoid, which is a simplified version of an actual human brain. So for my research, I'm using the brain cells and the brain organoids generated from the stem cell to study brain diseases. Ideally speaking, The best model system to study any brain disease will be the actual brain sample from the patient. However, the patient brain sample are available in very small number and are very difficult to obtain. So many scientists are using other model systems such as small animal as mouse to study the brain disease. However, the mouse brain is not exactly similar to the human brain, and the results are obtained from the mouse brain may not always apply to the human brain. Therefore, the brain cells and the brain organoids that are generated from the stem cell provide a very useful source for studying a variety of brain diseases, such as Alzheimer's disease and Parkinson's disease. For my research project, I'm mainly focusing on a brain disease which is called epilepsy. Epilepsy is a brain disease which is characterized by seizures in the patient. Epilepsy is caused by abnormal electrical discharges in the brain. Consider that our brain is like an electrical circuit. Sometimes there is a problem in electric circuit, maybe the two wires fuse, which can result in the flickering of light or the power failure. Similarly, in case of epilepsy, there is a problem in the electric circuit of the brain, which can result in seizures. Seizures are the brief episode where there is a reduction in the consciousness of the brain, which can cause the involuntary movements of the body. Without proper treatment, epilepsy can be a life-threatening condition. Epilepsy is a very common brain disease. It has been estimated that about 1 in 26 people in the United States will develop epilepsy at some point in their lifetime. So what are the causes of epilepsy? One of the main causes of the epilepsy are the genetic changes. The changes in the genetic material or the DNA of the cells can cause undesired effect. The DNA is like the blueprint of all the material in the cells. If there is any undesired change in this blueprint, it can begin to cause diseases such as epilepsy. The other causes of epilepsy may include trauma or injury to head, brain tumor, or the infectious diseases of the brain. For my research purpose, I'm mainly focused on understanding how the genetic changes in the DNA can cause epilepsy. So how do I do this? I do this by using stem cells. For this, I collect the skin cells or blood cells from the patient that have been identified with epilepsy. I convert these cells to induced pluripotent stem cells, and then I direct these cells to form brain cells and brain organoids. Similarly, I collect the skin cell and blood cell from a controlled individual. A controlled individual is roughly the same age as a patient and has no history of epilepsy. I convert the skin cell and the blood cell of the controlled individual to stem cells and then direct these stem cells to form brain cells and brain organoids. After this, I run a couple of tests, and I use these tests to compare the brain cells and brain organoid of the patients versus control. This helps me to identify what are the genetic changes in the patient cells and how these genetic changes may be related to epilepsy. So, for my project, I'm using the brain cells and brain organoids to identify how the genetic changes are causing epilepsy for modeling of epilepsy. In addition, the brain cells and brain organoids can be used for a variety of applications. They can be used for the testing of drugs in order to discover new drugs that can be useful for the treatment of epilepsy. Also, they can be used for testing different compounds in order to understand their toxicity. Also, scientists have come up with a method where it might be possible to correct the undesired genetic changes in the brain cells that have been obtained from the patients. After these genetic changes have been corrected, these corrected brain cells can be transplanted back into the patient brain, and they can replace the cells in the patient brain that have been damaged due to epilepsy. This is known as cell transplantation. Ultimately, all of this can result in the development of personalized treatment therapy for epilepsy patients. This can greatly improve the life quality of epilepsy patients. So, in case if you are more interested to learn about the stem cells and epilepsy, here are some of the resources. Thank you for watching this video. I hope that you learned something new from this video. Bye.